Today on High Tech Shower International, we'll bring you the second half of our special report on Brookhaven National Laboratory. In our continuing focus on biological research at the lab, we'll take a look at the world's largest database of three-dimensional protein structures. And then we'll introduce you to a researcher whose work is helping to fight bacterial resistance. And finally, we'll learn about some transgenic plants that may become new sources for oils. I'm Tom Dorick. All this coming right up. It was the early 60s. After nearly 30 years of work, Max Perutz and his colleagues completed the first three-dimensional model of a complex protein. The wire, plastic, and wood model required 16 feet of floor space and eventually garnered Perutz the 1962 Nobel Prize in Chemistry. In the early days of structural biology, researchers spent years attempting to interpret X-ray photographs. Some significant breakthroughs did occur, like Rosalind Franklin's work in the 1950s on the structure of DNA. However, determining the structure of large and complicated proteins proved to be much more difficult. Up until the 1970s, only a handful of three-dimensional protein models existed. But today, scientific journals are constantly publishing new protein structures. Also, our ability to manipulate these models has drastically improved. Just compare this early DNA model to one of today's complex DNA animations. So a lot has changed since 1913 when the first X-ray diffraction patterns were produced. But what's caused these rapid advancements in structural biology?
Molecular biology really helped a lot. So there was a lot of changes in sample preparation. It became much easier to prepare proteins, purify them. You know, you can do cloning, and so you get very pure samples. The whole data collection methodology has improved, and structure solution has also improved. All of this is coming around and helping each other in a synergetic way, and we've got this enormous growth in the field, which is putting a real strain on resources which provide information. But it's a very interesting challenge, really.
As we mentioned last week, the National Synchrotron Light Source at Brookhaven is an important biological research tool. Structural biologists from around the world use the intense X-ray radiation for protein crystallography. Such tools are another factor contributing to the growth of this field.
Yeah, is that 1, 3 oscillation? Oh, it's 1.5. Oh, that's okay.
The technical gains go beyond the simple instrumentation. Also, the other half of the story is software and the analytical procedures used to interpret diffraction data and ultimately end up with electron density maps of the structure you're looking at. So it goes hand in hand with this hardware developments as well as software developments that have made the whole process a lot more facile than it used to be.
Recognizing developments in this field, the U.S. Office of Health and Environmental Research is funding a new set of biological research stations at the light source.
So if anything, we're a growing presence here. We tend to be displacing chemistry and physics programs that are moving either to more advanced sources or losing their funding. So if anything, it's a growing proportion of operations here on the floor.
Besides being stunning images, three-dimensional protein models are keys to understanding subtle biological interactions. With a good model in hand, scientists are able to propose new ways of understanding fundamental biological mechanisms. Very often, the models generated by structural biologists can have a major impact on many areas of research. News of a new model can send shockwaves throughout the scientific community. Since a protein structure can radically change the way a researcher designs experiments, it's crucial that the scientific community has easy access to protein models. To fill this need, Brookhaven National Labs has maintained the world's most comprehensive protein database.
Basically what happened was in the early 70s, there was a feeling that this field was going to go very, very big. And there was going to be a need to store this somewhere so the rest of the community can access this information. And this was basically the birth of the Protein Data Bank. And this was spearheaded by Walter Hamilton from Brookhaven, who had the foresight knowing that this thing was going to grow. And it took about, well, we really started seeing the growth pains about 5 years ago. and we're now going through an exponential growth phase.
The Protein Data Bank is analogous to GenBank, the massive database containing all the published nucleic acid sequences. Both databases are free and accessible internationally through the Internet. However, unlike the one-dimensional DNA sequences available through GenBank, the Protein Data Bank contains three-dimensional information describing a protein structure. To make this information easily accessible, the Protein Data Bank established a World Wide Web homepage in August of 1994.
So if, for example, you want to find out about a certain molecule, whether structure has been reported and how it looks like, you would connect to our homepage and do a search. So, for example, if we're interested in looking at this particular entry, which is the acetylcholinesterase complex with tacrin, we could get the what it looks like and the Protein Data Bank entry. But you can also actually view this through a viewing program, and you could do this remotely through the network. And it fires up a viewing program, and so you see the molecule in there.
And through the World Wide Web, a user can immediately be connected to any other database that has information on a specific protein. And since accessibility is important, the Protein Data Bank can accommodate a PC-based viewing program for image manipulation.
We're getting about 1,000 entries a year now. We expect by the year 2000 we'll have 30,000 in the data bank. And the whole thing just exploded in the last 5-6 years, really, maybe 10 years. But really in the last 6 years, to the point that now we're getting about 100 a month.
These protein structures are not only of interest to academic researchers. Most major drug companies are experimenting with structured-based drug design, and several smaller firms like Agaron Pharmaceuticals and Vertex Pharmaceuticals specialize in this approach. Surprisingly, many of these companies have deposited their structures in the Protein Data Bank in a relatively expeditious manner.
Actually, it's a very unique experience, I think unique in the scientific field, in that both private and public sectors cooperate in a synergetic way. What I mean by this is, as you know, there's a lot of interest in drug design issues. And so there's a lot of interest within the drug community, the private sector, to access information that we have. They, in turn, actually have been forthcoming in depositing their own structures.
Although structured-based drug design has yet to show any major payoffs, it does help researchers focus their experiments. And there's no doubt that structural biology will continue to have a major impact on basic research. In our next segment, we'll meet a researcher who is using molecular imaging to probe some fundamental aspects of protein synthesis. Although much emphasis is placed on the importance of DNA, some scientists believe that RNA actually was the original storehouse of genetic information. Similar questions exist around the evolution of ribosomes. These ancient machines for protein synthesis exist today as large, highly complex mixtures of RNA and protein.
Now, most biologists believe that the primitive ribosome from which the modern ribosome arose consisted entirely of RNA. Because if you needed proteins to make the ribosome, and then you need the ribosome to make protein, it's a sort of chicken and egg problem, which is insoluble. But in fact, now what most people think is that the primitive ribosome was made entirely of RNA. But as life evolved, proteins attached themselves to ribosomes, which were very specific and which conferred better efficiency, greater accuracy on the ribosome. And that's why modern ribosomes all are intricate complexes of ribosomal RNA with many different proteins.
Understanding the complicated way ribosomes coordinate protein synthesis is one focus of Dr. Ramakrishnan's work. At Brookhaven, he has generated high-resolution images of key ribosomal proteins. His research also has important medical applications.
What's interesting about ribosomes is that a vast number of antibiotics act on ribosomes. And the reason for that is ribosomes are so old that the ribosomes of humans or mammals and the ribosomes of bacteria have diverged sufficiently so that drugs can act on bacterial ribosomes selectively.
Recently, much has been made of health problems caused by bacterial resistance. Spectinomycin, for example, was used for years to treat gonorrhea, but recently, several bacterial strains have developed spectinomycin resistance. With detailed protein structures, researchers may be able to understand resistance on a molecular level.
Our current dogma about how antibiotics work on the ribosome is that these antibiotics actually bind to ribosomal RNA, and they distort the ribosomal RNA, and the distorted ribosome is unable to function. Now what happens is you can now get a compensating mutation in a protein that binds near that region of the distortion that somehow accommodates the distortion, so that the mutant ribosome is able to function despite the distortion in the ribosomal RNA. So you've somehow compensated for it by introducing another distortion, which allows the original distortion to still work.
Dr. Ramakrishnan and his group have mapped some sites for antibiotic resistance. Eventually, they hope this will lead to antibiotics which can cripple resistant strains.
This is a cartoon or a diagram of the structure of S5, which is one of the small ribosomal subunit proteins. A lot of ribosomal proteins, in fact, most of them consist of these alpha helices, which are shown in red, and they're packed against beta sheets, which is shown as these green arrows that go up and down on either side. But the interesting thing is, this was the first ribosomal protein for which there was a good deal of genetic information. And if you see this loop that sticks out from the body of the molecule, it turns out that this loop actually has the sites for a large number of the mutations that confer spectinomycin resistance. And we were able to map them. They occur along this loop, and a number of highly conserved positive amino acids also occur on this loop, suggesting that this loop actually interacts with ribosomal RNA, which would be over here. This is the first case where genetic information about antibiotic resistance was correlated with the molecular structure of a ribosomal component.
Ramakrishnan's group has generated models of several other ribosomal proteins. The two shown here shed light on some fundamental questions regarding the structure and function of the ribosome. Another area of interest to this group is the packaging of DNA within a cell. Every human cell contains about a meter-long stretch of DNA. But this DNA is somehow packed into a nucleus only a few microns in diameter.
The problem is, how is this DNA condensed into such a small space, and yet at the same time allows the cell to have access to various parts of the genome that it needs? So this condensation is done in a very organized way, and DNA in higher organisms is organized as chromatin, and what we're studying in this lab are the details of chromatin structure and how that structure is modulated by various other proteins.
In general terms, DNA is packaged by creating helixes around helixes around helixes. One group of proteins, called histones, allow DNA to form more complex helixes. Ramakrishnan's group was able to determine the structure of one crucial histone called H5, and they were also able to determine how this protein binds to DNA. By analyzing this step in the packaging process, the research group hopes to better understand the control of gene expression. And coming up in our final segment, we'll meet a researcher who's engineering new types of gene expression in plants. All living organisms contain two types of fats. Stable, saturated fatty acids are composed of long chains of hydrogen-rich carbon atoms, while unsaturated fatty acids have one or more double bonds. For several years, Dr. John Shanklin has studied the conversion of fatty acids, and he has isolated important enzymes called desaturases in plants. Currently, he's working on manipulating the function and expression of these proteins.
The reason it's an interesting problem and the Department of Energy is interested is that it's an extremely energy-demanding reaction. A saturated fat is the most stable compound that we know of in biology, and to put a double bond in at an exact position along the acyl chain is very difficult. And so if you gave it to an organic chemist, he'd want to do it at 1,000 degrees with all types of catalysts, but here we have a protein that can do it on its own. And it can do it in a very specific manner, always in the same place.
Shanklin has focused much of his research on desaturases in plants. As humans began forming agrarian societies, we happened to domesticate similar types of plants. As it turns out, most crop plants that exist today have similar distributions of fats. As these plants convert saturated fats to unsaturated fats, they place double bonds in very specific sites.
So one aspect of our work is to try and take genes that encode desaturases that have unusual function from plants that are not crop plants, to take those and move them into an existing crop plant, soybean, canola, rapeseed, and to make a new crop that now the value of the oil is maybe 4 or 5 times higher than the oil that it already makes. So for instance, one thing we've got is this plant over here is every year we take 45,000 tons of soybean oil and we make a fatty acid that is made naturally in the seeds of this Spanish weed. And what we're trying to do now is to isolate, knowing about the biochemistry of it, we're going to isolate the gene from this, move it directly into soybeans, and we'll be able to simply extract this oil that we need from soybeans instead of having to take it and chemically process it. So we're making a renewable resource out of an existing crop plant.
In addition to moving natural desaturases into crop plants, Shanklin's team wants to design new enzymes that could create compounds that do not normally exist in nature.
So if we know how the enzyme grabs onto a fat, we know why it puts the double bond in a specific position, we can then tinker with the enzyme so that it binds it a little bit differently and puts the double bond in a different place. And in fact, we've already engineered a new desaturase that has never been seen in nature already, and we're in the process of trying to put that back into plants now.
Well, with that, we've reached the end of our special report on Brookhaven National Laboratory. We hope you've enjoyed this series, and thanks for joining us. I'm Tom Dorick. We'll see you next time.